laws, so something that Mendel didn't hit up on and a little bit difficult to see with his P studies, and this is very much the case for all of us because genetics aren't so 100% following Mendelian genetics. It's a beautiful background to give us kind of a basis understanding of what genetics is and what heredity is, but there's some little variations that I want to discuss. The first one is incomplete dominance. What incomplete dominance is you're telling me that your hybrids have a combination between the two parents. So it's not like you're either looking like one parent or the other. You're looking like a combined effect between the two parents. That is called incomplete dominance. Not one allele is more dominant than the other. So it's a combination between the two alleles. So here I can kind of see the red petal color denoted by uppercase R for all simplistic reasons. And here's white, seen with the lowercase r. And when mating happens, the hybrid is neither red nor white, but actually the heterozygote hybrid is pink, a combination of red and white, kind of mixing the two alleles together. This is called incomplete dominance. Another variation is codominance. In codominance, both alleles are expressed in the heterozygote individual. So it's not really more a blending of the phenotypes, more so than it is expressing those alleles at the exact same time. So here I have a white, denoted by uppercase W, petal color. Here I have a red, uppercase R petal color, and for those hybrids, it's actually spotted. We see both red and white, not pink, but both of those alleles get expressed at the exact same time. They're both dominant. This is called codominance. A beautiful example of codominance in us is our blood type. So if we're AB blood type, we're both A and both B. Those are both expressed at the same time. The last slide is basically showing you guys how Mendelian genetics is going to tie into our meiosis lecture. So beautifully sums up those concepts where we have the chromosome theory of inheritance. Really, we're talking about gamete formation, and we're talking about chromosomes being inherited by our offspring. And Mendel really paved the way for us to understand this. So how chromosomes behave during meiosis, how homologous chromosomes are going to line up during metaphase, how they're going to eventually split during anaphase, this all holds true with Mendel's hypotheses. So that really paves the way for his law of segregation and law of independent assortment. Law of segregation really telling you guys that alleles are going to segregate or separate. So that's seen here on the left side, and I can see that alleles are very much separating into their respective gametes. Over here on the right side, law of independent assortment, that dihybrid cross analysis really shows me that, where homologous chromosomes line up independently of each other. Let's say that this homologous chromosome wants to line up on the right side, and then this blue homologous partner is on the left side. Here, I don't see these pairs with blue being on the left, red being on the right. They're going to line up however they want. Maybe they'll line up that way, but in this image, it looks like the red chromosome decided to line up on the left side and the blue one on the right side. It doesn't matter how the previous set paired up. And so that's called the law of independent assortment. That's also going to give us this beautiful genetic diversity that we see in our population.